Hello. Today we'll continue our second lecture about cell signaling. We'll deal with cancer cell signaling. So cancer, in another definition, it is a wound that doesn't heal. There is a similarities in biological process between wound healing and malignancy. Cell proliferation, cell migration and recruitment, angiogenesis, inflammation, increased cellular metabolism, and many of the other of these processes regulated by growth factors as well. Growth factor may come from both malignant or normal tissues. The hallmark of malignant behavior consists of many characteristic features, like the ability to evade apoptosis, like the self-sufficiency in growth signal, like the insensitivity to anti-growth signals, like the limited replicative potential, like DNA damage stress lead to cancer, like oxidative stress as well, like mitotic stress, proteotoxic stress, metabolic stress, and the cancer cells can evade the immune surveillance and sustain angiogenesis, which lead to tissue invasion and metastasis. So it's a kind of dysregulation of the pH and aneuploidy and hypoxia and senescence and reactive oxygen species. So the growth factors in wound healing, you can see that growth factor like BDGF, it control the blood vessel closure, while EGF controls the proliferation and migration of epithelial cells, while BFGF, it controls as well the proliferation and migration but for the fibroblast, while the VEGF, it repair blood vessels. We'll start talking with one of the important class of receptors, which is called receptor tyrosine kinases, RTKs. The example we'll deal with is the Ras-MAP kinase pathway. RTKs have built-in tyrosine activity in their cytosolic domain. The ligands for RTK are soluble membrane-bound peptides and hormones. The examples like the NGF nerve growth factors and the PDGF, as well the fibroblast growth factors, epidermal growth factors, and insulin, there is a lot more. So initially the ligand when the ligand bound to RTK, it activates the tyrosine kinase, leading to activation of the Ras-MAP kinase pathway. These lead to the regulation of four main processes: differentiation, cell survival, cell metabolism, and cell proliferation. Some RTKs have been identified in studies on human cancers associated with mutant forms of growth factor receptors. Mutant RTK send a proliferative signal to cell even in the absence of the growth factors, so it is always switched on. Example, the active mutant form of the HER2. It is a receptor for EGF-like proteins. This enables uncontrolled proliferation of cancer cells even in the absence of EGF, which is required for proliferation of normal cells. So monoclonal antibodies, which are designed to target the EGF receptor, have proven therapeutically useful in these patients. So you can see here the receptor, and we'll see that the effects of oncogenic mutation in a proto-oncogene that encode a surface receptor. So these are it's a normal receptor, HER2 receptor normal, showing the exocytoplasmic part and the cytosolic domain. And you can notice that there is a crucial amino acid, which is valine, that help this receptor to bound to attach to the membrane. Once there is an oncogenic mutation that convert after translation the valine into glutamic acid, it transforms the hair receptor. It's transformed into new oncoprotein, and this leads to constitutive activation of the receptor tyrosine kinase and the dimerization as well. So it will be upregulating the RAS signaling pathway. So this is a pure example of converting proto-oncogene into oncogene. Another example you can see here, the EGF is inactive because it's not dimerized, there is no signal, Once there is a deletion of the exocytoplasmic domain, it is converted to a ERPP oncoprotein, and in this case it's still as well a constitutively active protein, tyrosine kinase. Either the two RTKs, once there is an oncogenic mutation, it will lead to upregulation of the signal pathway downward of this receptor. So this is a kind of ligand-independent receptors activation. So now we'll dissect more the Ras-MAP kinase pathway and see how the signal is transferred from the cell membrane into reaching to gene expression. RAS activation following a ligand binding to RTK induce a serine-threonine kinase cascade of reactions, which involve a lot of kinases, leading to ultimately to the phosphorylation of the MAP kinase and the transcription factors, which is recruited into the nucleus. So RAS, it's a GTPase switch protein, as we said, an example as we said from last lecture. It functions in transducing signal from many different RTKs. So here see, we'll take for example the receptor EGF. It waits for the signal of EGF molecule, the ligand, so it is still in a monomer state because it's inactive. And you can see here that RAS bound to GDP is in inactive form. Once the signal bound to the EGF monomer, it induce a dimerization of the receptors and autophosphorylation of the cytosolic domain of the protein. This phosphorylation recruits two important proteins, which are GRP2 protein and SOS protein. GRB2 protein have two important domains, an SH2 domain and SH3 domain. Once recruited to the cell membrane, the SH2 domain will bind to the phosphorylated tyrosine residues on the receptor, while the SH3 domain will bound to the SOS and localize it to bound to the inactive RAS. Soon, once the SOS bound to the inactive RAS, the RAS is now got activated through the displacement of the GTP into a GTP. Once RAS is active, it will transduce the signal from the cell membrane into the cytoplasm. So the active RAS will start to work on the inactive RAF. Inactive RAF is in the cytoplasm and is inhibited by bounding to another protein called 1433. This protein bound to the RAF and inactivated. RAF have two important domains, an regulatory domain and the SC terminal kinase domain. Once the RAS is active, the RAF is recruited to the cell membrane, and the RAF is now active by binding to the active RAS. Once RAF is active, it will induce the activation through phosphorylation of MEK protein to be active MEK phosphorylated protein. This active MEK will activate the MAP kinase through phosphorylation, leading to active MAP kinase. This MAP kinase will soon dimerize and will activate another protein to facilitate the recruitment and transcription factors, to facilitate the recruitment of this protein, the transcription factors, to the nucleus. So the MAP kinase regulates the activity of many transcription factors, controlling early response genes. So what's early response genes? GF addition to a quiescent culture mammalian cells at the G node or G0 causes a rapid increase of the expression of 100 different genes. These are called early response genes because they are induced well before cell enters the S phase. The important ERG encodes the transcription factor C-Phos and C-Gen, two of the most important transcription factors that control the cell cycle. Both transcription factors induce expression of many genes encoding protein necessary for the cell to progress through the cell cycle. The enhancer actually its enhancer is a sequence on the DNA that regulates the CFOS gene contain a sequence of nucleotides called serum response element. So for your knowledge, any protein that needs to bind on the DNA need a specific sequence on the DNA, especially if it's a transcription factor, need a specific sequence in the DNA which is called serum response element, SRE. Like you can see here. So CFOS gene have an SRE upstream of the coding sequence of the gene. So this SRE serum response element is activated by many growth factors in serum. These enhancers contains DNA sequences that bind multiple transcription factors. Some of these transcription factors are activated by the MAP kinase. We just described the MAP kinase. Others by different protein kinases that function in other signaling pathways. So you can see here we stopped at the MAP kinase, which is activated by MEK kinase. So soon after activation, the MAP kinase is dimerized and activate the inactive P90 RSK by phosphorylation to form active P90 RSK. This as well, the dimerized MAP kinase will be recruited inside the nucleus to activate the TCF, it's a transcription factor, by phosphorylation as well. So both the TCF and the active P90 risk is now inside the nucleus, so the activation of the phosphorylation of P90 RSK leads to its recruitment inside the nucleus. Once it is recruited inside the nucleus, it will activate another transcription factor, it's called SRF. So now we have two transcription factors, TCF and SRF, are now activated. Once both are phosphorylated and activated, they'll bind to the serum response element of the CFOS gene and induce expression of CFOS gene. And this is the ultimate end of the initial signal that came with the EGF ligand when bind to the RTK. So now the signal have been conveyed from the cell membrane through the MAP kinase pathway until the expression of the CFOS gene. Now we'll talk about another receptor that is important in tumors, which is called PDGFR receptor, platelet-derived growth factor receptors. This platelet-derived growth factor receptor, normally there is isoforms of this receptor. So isoform BDGF-A and B, it's a signal that leads to autocrine stimulation, self-stimulation. This BDGF A and C, another variant C with A, lead to the recruitment of cancer-associated fibroblasts, which once they are recruited, they secrete another factors that induce more activity of the initial cells that secrete the BDGF A and B. This cancer cell, by the secretion of VEGF, will induce angiogenesis for the tumor. These plus the signals coming from the cell, the tumor cell BDGF B and D, another two isoforms, will stabilize the blood vessels and lead to the deattachment of the pericytes. Pericytes, it's a type of cells that aligns the blood vessels. By these two processes, the cancer or the tumor have succeeded in sustaining angiogenesis. Another factor, BDGF-B, will induce one of the important mechanisms of tumor for metastasis, which is epithelial mesenchymal transition through the autostimulation of the TGF-beta, tumor growth factor beta. This will lead to the metastasizing. You can see that the cells with the red nucleus start to metastasize away from the tumor, and once reaching a blood vessel, it will reach all over the body. And as well will recruit more cancer-associated fibroblasts to all places in the body. And this will lead to invasion and metastasis. You can see here that there is a lot of growth factors involved which make the tumor succeed in self-sufficiency in growth signal. So the PDGF there have been numerous mutations and alteration of this receptor PDGFR in a number of tumors. For example, if there is amplification in the gene, will lead to, you can see that you have like 6 copies of the receptor on the cell membrane. This is obvious in the glioblastoma, which is a brain tumors. While in other cases, if there is a point mutation in the cytosolic part of the PDGFR alpha, you can see that these have been discovered in the gastrointestinal stromal tumor. While if there is a hypereosinophilic syndrome, where there is a dilation in a region of the PDGFR beta that doesn't enable this receptor to be expressed on the cell membrane, will lead to hypereosinophilic syndrome. In another mutation, there is a translocation between 2 genes, leads to a receptor that is a mix of 2 genes, came from the translation of 2 different messenger RNA, or a gene that have been translocated, and this obvious in the myeloid leukemia. For epidermal growth factor receptors in tumors, these receptors composed of 4 families of receptor: EGFR, HER2, HER3, and HER4. In normal cells, 20,000 to 200,000 molecules of EGFR per cell, while in cancer, this number increased to 1 million molecules per cell. So what's a HER2 receptor, human EGF receptor 2? It is overexpressed in breast and ovarian cancers, 25% of the cases, as well is closely associated with increased metastasis. Its overexpression. It induces a transformed phenotype, and the upregulation of HER2 receptor leads to MAP kinase hyperstimulation. And as we said before, MAP kinase hyperstimulation lead to more transcription of the CFOS and CJON, so the cells will keep on cycling and divide. A point mutation valley into glutamine will convert the normal HER2 into the new oncoprotein, as we have just said early in the lecture, which is initiator of certain mass cancers. These mutations enables HER2 to dimerize and is constitutively active in the absence of the ligand. They found that a monoclonal antibody specific for HER2 can block the activity of the HER2, and successful new treatment for a subset of cancer, of breast cancer, that overproduce HER2. And you need to know that not all breast cancer have overexpression of HER2. HER2 antibody is injected into the blood and recognize HER2 and cause it to be internalized. So this will selectively killing the cancer cell without any apparent effect on normal breast and other tissues. So you can see here from a Western blot, you can see here the difference between 2 MCF7, these kind of breast cancer cell lines, but the MCF7 HER2, it have an overexpression of HER2 receptor. So you can see both of them have a MAP kinase, while the one with the HER2 overexpressed, you can see this one have only the phosphorylated form of MAP kinase. This proves that the upregulation of HER2 will lead to the activation and the phosphorylation of MAP kinase. And of course, actin here is used as a loading control to make sure that you loaded the equal amount of protein per each lane. So for the vascular endothelial growth factor receptor VEGF in tumors, you can see here normally the tumor is inside, is not have been metastasized yet, and you have the bloodstream and the represented by the blood vessel. You have the endothelial cells, you have the pericytes. So the tumor itself will start to secrete tumor-derived VEGF, vascular endothelial growth factors. This will lead to formation of the blood vessel to reach the tumor, and through the expression of the receptor as well, that the VEGFR1 and 2, and the action of the growth factor released from the tumor lead to the formation, as we said, to the blood vessels. Soon after that, the lymphatic vessel as well have another receptor VEGFR3, which is the third isoform, which is active as well when it touches tumor-derived VEGF. All of this will innervate, ensures that a tumor will have angiogenesis and the lymphatic circulation. So what is the therapeutic target for the VEGF signaling? Normally in the market now, if there is three different strategies to deal with tumors that secrete VEGF. You have the IRSA, which blocks the production of VEGF from the tumor, and there is another strategy which is the SUTENT, which blocks the receptor, the VEGF receptor, from accepting the signal, which is the VEGF ligand. And you have as well the Avastin, which neutralizes the signal in the bloodstream. So these are the three different strategies are needed to treat the cancer derived from the VEGF secretion. FDA approved Avastin for metastatic colon cancer and non-small lung cancer in 2004. As well, in 2008, it was approved for breast cancer. So is Avastin is very good? No, it have a lot of side effects. Some of them, like impaired wound healing, and is very obvious because wound VEGF needs its main function is to make blood vessels. So if you are taking Avastin, any wound you have, it will not heal. It will lead, of course, to bleeding. It would lead to hypertension. It would lead to bowel proliferation because it's affect as well lymphatic blood vessels and blood vessels in going to the bowel. It will lead to CNS capillary leak. And it will lead as well to something called nasal septum perforation. And you can see here the septum is incomplete. There is a perforation between the two chambers of the nasal chamber. So you can see here, this bar chart shows over the years up till 2007, the amount of transdermal drugs used for cancer have been approved. And you can see it is increasing through years. Here we'll see an example of a cancer. It's a bone and how bone metastasize. It's a result of an integration of many growth factors and their receptors. So you can see here the primary tumor, once again, detachment and angiogenesis leads to its migration. It will be a metastatic cancer cell. This metastatic cancer cell will secrete osteolytic factors that lead to the activation of the osteoclasts. And the osteoclast, actually, it is a macrophage of the bone, which lead to the eating of the debris, and it's like immune cells inside the bone. These osteoclasts will soon secrete more bone-derived growth factor, will activate more metastatic cancer cells to secrete osteoblastic factors. These osteoblastic factors will upregulate the formation of the osteoblast, and osteoblast actually the kind of cell which secrete a bone. This lead to more maturation, and more new bone is formed, and the larger number of cells is formed. This lead to under as well the physical factors like hypoxia, acidic pH, and extracellular calcium. This process is upregulated and enhanced, leading to more cells, and this lead to bone cancer. Nuclear receptors. The nuclear receptor need a signal which is small lipid-soluble hormones like steroid hormones, retinoids, and thyroid hormones. Signal can diffuse through these lipid-soluble hormones or steroid hormone. They can diffuse through the plasma and the nuclear membranes and interact directly with the transcription factor they control. The intracellular receptors, which are the nuclear receptor, function as a transcription activator when bound to their ligand, which is the signal. Here you can see an example of 5 different nuclear receptors, and you can notice from the bottom figure, which represented the general primary structure, that a minimum they need to have 3 different domains: green, blue, and violet or purple. These 3 different domain or 3 different functional difference domains they share, all the nuclear receptor, most of the nuclear receptors share these 3 domains. We have a DNA, we have a variable region domain, we have a DNA binding domain, and the ligand binding domain. This variable region of domain may function as an activation domain, while the DNA binding domain has a repeat of C4 zinc finger motif that helps the protein or the nuclear receptor to bind to DNA. While the ligand binding domain, once there is no ligand, it acts as a repression domain for the nuclear receptor, for the nuclear receptor. So looking closely into the response element of this receptor, as we said, the response elements are a nucleotide sequence on the DNA act as an enhancer that need to be bound by the transcription factors. So every kind of a signal, every kind of signal like steroid or thyroid hormones need a response element. So you can see here the first two, which are the response element of the glucocorticoids and the estrogen receptor, have 6 base pairs of inverted repeats. You can see that the direction of the sequence is the same but inverted and separated by N3, which is a 3 base pairs. For example, the steroid hormones receptor bind to DNA as a symmetrical homodimers, so it needs a 2 identical molecule to bound to that DNA. While in this case, for the vitamin D3 and the thyroxine response element TRE and retinoic acid response receptor response element RARE, you can see that the response element is different. You can see that they are actually direct repeats of direct repeats, so the sequence is direct, not inverted, separated by either 3 or 4 or 5 nucleotides, base pair. So these ligands, so the proteins that need to bind to this response element had to be heterodimer. And this heterodimer either RXVDR or RXRAR in case of different signal, either vitamin D or the retinoic acid. The specificity for responding to hormone by binding distinct receptor is determined by the spacing between the repeats. So you can see the spacing, the ends in 3, 4, 5. Actually, it's important in defining the specificity of binding. So the hormone binding to a nuclear receptor regulates its activity as a transcription factor. The mechanism that hormone binding controls activity of nuclear differ from heterodimeric and then homodimeric receptors. So for heterodimeric receptors, it's located exclusively in the nucleus, so these receptors are in the nucleus. In the absence of their ligand, they repress transcription when bound to their DNA response element by a process of histone deacetylation. It's a mode of epigenetics at nearby nucleosomes. In response of hormone, this ligand binding domain of RAR monomer undergoes a conformational change and the direct histone hyperacetylation at nearby nucleosomes. That's why to open up uncoil the DNA for transcription. While the homodimeric receptors is found in the cytoplasm in the absence of their ligands. Once they're bound, these receptors are recruited into the nucleus to bound to the DNA. Here is a simple experiment to prove that there is a hormone-dependent translocation of the homodimeric glucocorticoid. So that means the glucocorticoid receptor only translocate into the nucleus once it's bound to the ligand. And also this experiment will prove that it needs the hormone for the translocation. So in the first figure, you can see that the top figure, it cells that not treated by DX, and the second one is cells treated by the DX. DX, it's a signal, it is a signal, the ligand actually. So the second cell is treated with the ligand, which represent the glucocorticoid, while the top figure is not treated, the cells is not treated. So here, what they actually done, they have the cells express a kind of protein called the beta-galactosidase. This only beta-galactosidase. So beta-galactosidase is like a marker. So they can, by the fluorescence of beta-galactosidase, they can know the location of the protein. So you can see this beta-galactosidase, it doesn't matter if there is a ligand or not, DEX or not, that this protein stays in the cytoplasm, because it's brighter in the cytoplasm. So it is the location is expressed or monitored by the brighter color. So this protein is localized in the cytoplasm no matter there is a DEX or not. So in case if the protein, this beta-galactosidase, is fused to the glucocorticoid receptor, and this glucocorticoid receptor, as we said, it's a homodimer, so in inactive state, no ligand, it will stay in the cytoplasm. So you can see in the first figure, when there is no DEX, no ligand, this protein bound to beta-galactosidase, which we have said that it is a marker. So you can see the brighter color is in the cytoplasm. Once we put DEX, which is a ligand, this protein is translocated from the cytoplasm into the nucleus, because the brighter color now is inside the nucleus. So that means it is only translocated in the presence of DEX, which is a ligand, which mimics the glucocorticoid. In another experiment, they thought that they want to think which domain, as we said before, there are three different domains: a regulatory domain, a DNA binding domain, and the ligand binding domain. So they took only the ligand binding domain and fuse it to the beta-galactosidase and do the same experiment. They found that this part of the protein is translocated as well. So that means the domain which is responsible for the translocation is the ligand binding domain, because you can see it is moved from the cytoplasm, the brighter color is moved from the cytoplasm into the nucleus upon the treatment of DEX. So you can see here in this sketch, in the absence of the hormone, GR is the glucocorticoid receptor is anchored in the cytoplasm, complex with an inhibitor protein, one of the major heat shock chaperone protein. So you can see here in the figure, the inhibitor protein, heat shock protein 90, this is used to inhibit the GR glucocorticoid receptor, and you can see this GR have three domains: the ligand binding domain LBD, the DNA binding domain DBD, and the activation domain AD. So once there is no ligand, there is no hormone, this GR will stays in the cytoplasm and bound with the inhibitor HSP90. As long as the receptor is restricted in cytoplasm, it cannot interact with the target genes. It cannot be translocated. So no activation of the transcription. The hormone binding, this hormone, which steroids, which is internalized and bound, this lead to the release of the inhibitor, and now the glucocorticoid can bound as a homodimer. You can see that both molecules are the same, and they bound on the response element for that, which is called GRE. Glucocorticoid response element. Once a receptor hormone binds to response element, it activates, of course, transcription by interacting with chromatin remodeling and histone acetylase complex and the mediator to open up the DNA for transcription. Thank you, and this concludes our lectures of advanced cell biology. Thank you.